Hello, everybody. Welcome to the Basscast. Today's episode will be about some of our observations that we've seen through the Discovery Farm up to this point of the year before harvest. But before we get going, we want to introduce Heidi up there on the screen. Heidi, why don't you introduce yourself and give us a little bit of some background and what you got going on?
Hello, I'm Heidi Beckendorf, and I'm currently a first year student at Ridgewater, and I'm doing an internship through Bass for marketing and communications. I'm currently going for my egg business degree with a certificate in marketing communications, and I hope to market for an egg company in my future.
Awesome. Well, welcome to the team. We're happy to have you here, and hopefully we got
Yeah,
some good stuff for you to run with.
yeah, no, welcome, Heidi. We're looking forward to working with you the next 4 weeks. So hopefully you learn a lot and help us out with what we got to get done here during harvest time. Help us get some promos done for what yields are doing and things like that. So
Yeah, thanks for having me.
sure, it's our pleasure.
All right. So today is Friday, October 10th. We are not that far off from harvest of the Discovery Farm. Well, one is what's our guess as to when we'll be in there with the combine?
As of right now, Andrew, we're going to start with doing our strip trials. Like where the seed treatment trials were and our skip row work with conventional combine or a small combine, but not one of our actual plot combines, and a weigh wagon.
Okay.
Set strip trials. And so we'll be harvesting, uh, grab that information as soon as we can on the seed treatments and the skip row works. We can start putting that information into our book now. Like if we have skip row friendly corn, your preferred for that type of effort, that's what we're doing. And then to understand if we had yield benefit or not from these seed treatments. So.
Oh, now what's our guess right now on October 10th at what like our field moisture is out there?
I'm going to say Maria's right around 19%
Okay.
moisture base because we've been harvesting another field that was planted, just a little bit before that, and it's running somewhere between, uh, gosh, 18 and 21.
Okay, so getting close.
Yeah, that's spot on for us up here. I mean,
So
we wait much longer, we're going to miss the season.
right,
So,
right. So we have a bunch of different experiments, trials, different things going on. And I guess the main point of us having the chat today is to look at some of the observations and what we've seen up to this point, and then come back post harvest to kind of match what the results were to those observations. So I guess, why don't we run through again some of the different things that we have going on at the Discovery Farm? And Travis, you can start by kind of just giving a descriptor of the different things that we have going on out there.
Sure. So we do have a lot of skip row work planted right next to that same hybrid with the same inputs at a higher population without the skip row. So we're going to be able to see the side by side what the yield difference is going to be. We also have the show plot that's right across from that with all of the different hybrids that had AEA products put across it. Then the middle of the field is going to be 3MG trials. And then at the very back is our seed treatment trials, where it has our Maria hybrids, our hybrid with multiple different types of seed treatments to see how the responses go.
So in the skip row, did we adjust population, or did we just remove that like a third of the seed from that plot?
we removed a third of the seed. We just turned the row unit off.
Okay. So we're not,
So,
there's no bump in the other two if we're so okay. Just so
yeah.
so people understand what we're talking about.
Yeah,
Mhm.
we want, part of it is self-serving. We want to understand the flex in a hybrid too,
Sure.
because they need to flex if they're going to work in this situation.
So
And
let's go from there then. The observation in that skip row, are there any hybrids that we have in our lineup right now that you're seeing or that you're surprised by, or one initially right now that we're thinking is, or that a group of them that are really geared towards working in a system like that, whether it's skip row or 60 or something that is, not standard 30s?
Well, Travis, if you made some observations, I've got a few that I
Okay.
can, but go ahead.
So of course we just know from observation in the past that Vanessa is one of the hybrids that works well in a skip row. Then I was very surprised to see how well Kit is responding to a skip row. And then Dot, I've noticed that our Dot is putting out a much larger cob with bigger seed in a skip row. So it's giving us a better idea of genetic potential. We've given the plants a whole lot more light. So just physically, they look different on top of increased airflow. So we're also seeing less incidences of fungal disease.
Yeah, and I really like the difference in the corn plants from the standard 30-inch row versus the skip row. You know, we've got bigger stalks and the ear size is definitely larger across all hybrids. But uh, I think my one disappointment was is I didn't see as many two ears out there as that I saw like when I was in Kazakhstan at Armin's dairy farm. And he was here and looked at it last week with me, and he said that our soil isn't quite ready to help these hybrids perform like that yet. They've got to get more biologically fit,
Okay.
is basically what he determined. Because he said for him doing the skip row work, as his soils got more biologically active, the bigger the plants and the bigger the ears that he put on out there, or more number of ears.
Sure.
And his commentary was is Duchess and Vanessa were probably the best ones that he saw, but he doesn't have access to all the hybrids we have in our lineup either, because we have some that can only be sold in the US.
Sure.
So, but one thing that I'd like to follow up on after we harvest is how much the cover crops grow after harvest between the two areas as well, because the skip row has quite a jump on with the cover crop growth versus the standard seeding pattern. And so that's another thing I'd like to monitor, because some of that would be, you know, I don't know if it's a biomass rating we take or something like that, just to understand what there is. And then when I walk through the various hybrids, you know, it's a matter of how much light penetration we got to the ground for how much we have established in the solid seeded. And so I I think I can say today that the more upright leaf material and the shorter plants had more growth in the cover crop. And yeah, that's kind of a no-brainer statement. I get that.
Right.
But I mean, that's the observation I have right now. But if I take that same look or that same idea and I go to the skip row, they had no advantage being upright leaf that I can see right now. You know, the upright leaf didn't have more cover crop growth in the skip row versus the more flatter and wider leafed products. And so that's my observation as of now on that.
When
So-
we started the program there, the project at the Discovery Farm, we took soil samples and ran some tests. Travis, what was the the method? What do we do? Where did we send it? And what did we see when we sent that stuff out?
So the first test we took was end of winter, beginning of spring. We sent that into Biomakers and got a DNA test done as well as a Haney. So the idea is we wanted to create a baseline before we started putting inputs on the farm to kind of see where it was at. And we were very surprised by just how poor some of the ratings came back. So we were very low in fungal diversity. Sometimes we're at 1 part bacteria to 200 parts or more fungus. So we are lacking there for sure. Solubilization of nutrients that wasn't really there. So we had a lot of things tied up and lots of pathogen pressures, especially in our low spots. And so we knew going in, once we got those results back, that there was going to be a challenge on the farm. And once we started planting and putting out these biological products, we wanted to make sure that we had tests in and received the results back before our field day, which was on August 28th. So when we got the results back, what we found was our fungal levels had raised substantially to the point that it averages out about probably 1 to 40 now. Now, there's a lot of reasons that could be. That could be due to fungal release. It could be to when we were taking the tests. It could be creating a microclimate around the soil zone by having cover crops out there that are going to support fungal growth. So there's a lot of things there that we're still kind of trying to figure out, and a lot of solubilized nutrients. So we saw that the plant health was doing really good. So now it's just a matter of kind of keeping an eye on where it's going to be in the future. So we're going to continue to go out there. We have GPS coordinates from where all these tests were taken. We did it simply by splitting the farm into 4 quarters, taking samples in each quarter, one from a high spot, one from a low spot. So now we will continue to go back, take tests, and kind of watch the progression as of the farm as it continues in the regen space.
So, and a little bit more background on the at the beginning, Andrew, is that this farm had been no-tilled 3 years before we got it, and we continued with no-tilling on the farm. And the young farmer that was farming it before us, he decided to exit farming, but he was farming that farm exactly the way Big Ag would tell him. And the other thing is when I saw the list of diseases that were prevalent out there, that made sense based on the genetics that he was planting. What would, those particular diseases, like those genetic backgrounds that he had. And so I guess for me, it was confirmation that these tests are real, that we're getting good information because it tracks with what we know, let's say, from hybrids backgrounds and what they would be predisposed to for disease. And we found it in the soil. It's there. I found it very interesting how these soils migrate, their biology to fungal ratio. Actually, we're looking at fungal to or to bacteria, and just incredible differences. And Travis will get all this highlighted here for us when we put together a report exactly how these things are changing, because we'll take another test again after harvest on this farm. So just so we understand what's going on, because part of the whole idea for this farm is how fast can we take a farm from Big Ag style farming to regenerative ag.
Sure.
And get these soils to let's say the microbial level that we need to really push the boundaries for top end yield.
One thing that I noticed at the field day was in areas where we had cover crop, and this farm had never had cover crops on it previously. So the areas where we had cover crops and then where we didn't have any, where we didn't put any in, the difference in the smell of the soil was really, really eye-opening. Where the cover crops were, the soil smelled kind of what it should smell like, and where it didn't have cover crops, it was kind of almost foul smelling.
So here's an interesting tidbit for you, though, Andrew, is that when Armin and I were out there walking with the Kazakhstan farmers he brought over, when we dug in the center of the road, we had no cover crops. That was the blah smelling soil. There was nothing, you couldn't really smell any life in it. And we all kind of remember from kids what fresh soil smelled like after plowing.
Right.
And that wasn't there. Taking a spade and turn it up, we just didn't get any soil life smell. But when we dug next to the roots on what we consider our biology friendly hybrids, when you were right next to the root, you could smell that.
Yeah.
So something's happening already. But you get out a little bit more into the bulk soil, let's say, 4 inches away from where the brace roots are at, you start to lose the smell. And by the time you're 6 to 8 inches out, you can't smell it anymore. It was incredible. And so that's one quick way that we can test hybrids to see how biology friendly. by what they're doing, because there's some hybrids you dig by and you can't smell anything.
So, within our lineup, are there any that you're noticing have a better response to that? In what that observation was that you just your nose observation?
So I didn't go out and do many hybrids. We did the ones that Armin had. So it would have been
Sure.
basically Vanessa and Duchess and to smell that and then go between the rows. Oh, but by the way, though, when we did go where the cover crop was at, between the rows we had that smell. And it wasn't necessarily stronger or weaker than by the corn plants then. So the cover crops made a tremendous difference on perking up the life of that soil.
Just having
And
those roots
by
in
the
the
way,
ground is pivotal.
repeat that. I'm sorry.
Having the roots in the ground all year is pivotal.
It's huge. It's huge. And so the other thing I'll mention is our cover crops were planted at V3, and I think we need to go earlier than that. And based on some of the information we're picking up from talking to guys who've done this a long time over broad acres, they think we should be able to do it, V1, V2, or even at planting. And so that's one area we'll probably look forward to next year. And
So
then
we'll see when we get the combine in there if we have any drag from competition of any of that stuff. But from what I've seen is that with the right mix of stuff in the cover crop mix, that we're not taking away from the corn plant, and in fact we'd probably be more beneficial or adding to the entire system, all being a positive.
Yeah, the diversity helped. We did try and pick things that we thought would work best with corn based on our discussions with agronomists at Green Cover, because I kind of told them what we wanted and what we wanted to do, and then we went through each addition to the mix to say that we
Right.
thought would be the least competitive was corn, but give us the broadest diversity between the rows. And so far, that looks like we did fine. In visiting again with Armin, he would like us to get products that are taller and produce more biomass than what we did. And his logic was is that as soon as you get that first killing frost, they melt. And we went with the shorter ones because I didn't want them to get in the way of our corn harvest.
Sure.
Mhm.
And so the jury's still out because I think it's going to take a lot colder frost to kill them where we live here than I want to wait for to go out and combine.
Right. Well, even
Because
in me where I'm at geographically compared to you guys, where it's going to be a little bit different, where that would be, it could be a month later than what you guys get up there.
Mhm.
So
Exactly. And I think his killing frost comes a week or two earlier than ours does. And so that works for him. The other thing is why we don't want to wait is because I want to catch that November 1st deadline for planting cereal rye. Because we're going to go back out there and plant cereal rye again because I want to keep building that mat. Because the other observation we had out there is where we had good residue either from the cereal rye that I had out there previously or where I had more residue from a year or 2 years ago from being no-till when we scraped away that residue and then smelled the soil, it was a lot better there for the aroma from the soil
well,
versus a bare spot.
and then this year going into next year, we'll be in a better spot than we were a year ago when we had taken over the property and then got, because when was that planted, Ed? That was
So last year it was planted about the 15th of October, the cereal rye.
right.
But it was so dry, we needed a rain to bring it up, but we didn't get the rye or the rain until probably the 10th of November.
Right.
And so it grew from about the 10th of November till about, let's say, the 3rd of December. And then it didn't come up very well around the field, and you could especially see it on where the end rows or border rows around the field where he must have made a double pass of herbicide to control the weeds in the headlands, and we didn't get hardly any cereal rye established in that area.
Sure.
And then since the winter was open, we had a lot of winter kill of the cereal rye in the soybean side. that we've had, because our east side was soybeans, our west side of the farm was corn. So we did corn on the whole farm. So we had corn after soybeans and corn after corn.
Right.
And so we had less of a stand on the soybean side because there was at least more residue on the corn side with the cereal where I had a better chance of surviving, but it still was probably less than 70% on where we had the corn stubble.
So with the cover crops that are out there right now, and we have them interseeded against in every hybrid that we have in the lineup, are we noticing that some don't mind it at all, or thrive in that system, and some, or are there any that we're saying, hey, do not put into a system where we're operating with a cover crop or an interseed program?
I think we'll know that for sure after the combine harvests the research plots because we have the exact same trials with and without cover crops out there. And so that'll tell us for that. But you can definitely see some hybrids don't mind it, and other ones are showing some stress from it. And I'm 95% confident that the combine will show us that in black and white.
Right. Oh.
Yeah,
so now in our seed treatment trials and our early observations pre-harvest, Travis, can you kind of go through what some of those observations are and see what type of, different treatments that we've run or different programs and how those are responding up to this point? And then we can follow back up, after harvest to see if these observations up through the year to this point match with what the combine is going to tell us.
Sure. So very early on, once we got the corn planted, we saw obvious differences in the size of plants that were coming up and the thickness of the stems, basically across the board, with all of the different treatments that we put out there. And we kind of kept tracking that throughout the season. The plants seemed like they were much healthier. Going through, we have AEA product out there called BioCoat Gold with their seed flare. Those plants looked excellent. We go out there, we take samples of the ears as they set, and we're seeing less incidences of fungal infections. We have Mycobac out there. And we had Willie come out and gave him some samples of the roots, and he took them back to the lab to check to see what the mycorrhiza colonization was, and we were confirmed to have substantial mycorrhiza growth on those roots. And then we have like the Newfields product out there, which is more of a nitrogen fixer. So there's lots of different angles that these companies are taking with the bacterial and fungal teams that they're putting on out there. And generally across the board, we're seeing a positive response. But then the question becomes, how do we start mixing these things together to create a kind of the pinnacle of being able to unlock the potential of the plant's genetics? And that's kind of where we're coming out there and looking at even our own seed treatment and going like, what changes can we make to this? But generally, it's quite amazing to see how these seed treatments have affected the plant health, as well as it looks like the yield as well. We'll know once the combine goes through the field.
So, in my observations, just walking fields east to west, is more things that had a biological seed treatment towards the end of the year looked significantly better than products or hybrids, ours or otherwise, that had a standard chemical seed treatment on it at plant. So I think that we were setting that plant up from the beginning better in that system with the biological seed treatment and different products like that to help it finish really, really strong. Now, some of the observations I've seen, it might have taken a little bit longer to get going, but it definitely caught up and is now surpassing anything else that I've seen by competition. So speaks a lot. Oh, now, if we're looking at the, what we know about hybrid to hybrid and what we think would work in those skip rows, is what you guys seeing kind of in line with what we talked to farmers about, where kind of right? Especially some of the ones that we work with that we say, hey, these are really good for, low population situations. Like Vanessa, right? Vanessa's showing out pretty well in the skip row. That's one of the ones that we're going to talk to guys about. If the maturity zone, if it fits in your zone, plan it at this population, and we'll really be able to express that flex. Is there anything that kind of jumped off, through at this point of the year, and you said, oh well, now we know that this one has maybe more flex than we thought it did, or less?
Nothing stands out in my mind of being an absolute no. We made the wrong recommendation or anything like that. I think we did good for the spring just based on our population study work that we've done with these hybrids. Because in our minds, the assumption we made was that if they can maintain yield with lower population, they should work, they should have high flex then to work when you go into the skip row. Now, if it's skip row or 60-inch row corn, there's going to be a difference there too. And so there's a couple other figurations maybe we'll look at in another year where we do some 60s or twin 60s, or, I got to figure out how we do all that. But there's other options that maybe going forward we can play with.
Well, now, like what you said with Vanessa, and where you and I had seen her in Northern Illinois, Ed, and a little bit different, but we were seeing a lot more doubles, and some 30ers that were out there, and so it's a little bit different, right, from what we're seeing at the Discovery Farm. So then the question is are there in Northern Illinois, is Greg's soil just that much better than what we started with at the Discovery Farm, and we can work towards that, or is it I guess we'll see some of the data when it comes in from there. But
Well, I think at Greg's place there, he's so far ahead of us with his soil health than what we are on this farm.
yeah,
And so then I saw some of the same characteristics on his farm with Vanessa that I saw on Armin's farm in Kazakhstan. So that lended the credibility to Armin's statement when we were talking based on what I saw at Greg Thorne's place. And so that it's kind of fun when you can put these pieces together, what different people say from different parts of the world.
right,
And because one thing Armin told me over and over during the 5 days we spent together is that you don't know how much genetic potential you have in these hybrids yet. Because he's seen his yields go up each year as his biology got better. And since our hybrids like biology, they're attracting more and more biology, even more so than what we're applying or what he's applied. And I think, Travis, you can kind of have a little testimony to that when you're checking Maria out at the farm and you went to the nursery. We didn't apply any products, and you can tell us what you found there on Maria.
So when we went out and tested the Maria, we found that there was some native mycorrhiza growth out there. We also found some biocontrol agents that nobody has put out there. So there's natural components. I mean, this farm is not that far from the Discovery Farm, but just even the conversation when it comes to endophytes, right? It's like biology living inside of that seed that you're purchasing. That's going to be in there whether it's intentional or not. So just by where you're sourcing your seed from, if you're putting a cover crop out there, if you are using different species, that's going to influence that soil in the long run because there is biological carryover from one year to the next. So it's very interesting to be taking all of these tests and looking through all of that data and doing side-by-side comparisons and just realize how drastically different your farm can be, even if they're just right down the road from each other, just based on farming practices.
sure.
So we're going to have to kind of pull that apart because ultimately, we want to be able to recommend biology to people and fit it to their farming practices. So if you're a guy out there tilling and putting on heavy nitrogen, then we're going to probably want to rely on these resilient microbe teams that don't mind that type of pressure. But once we get into trying to produce plant hormones and trying to use biocontrol agents or mycorrhiza, we definitely have to be better stewards of the soil so we can go out there and have this carryover from one year to the next.
Well, based off of what we just talked about with the differences in our field there at the Discovery Farm, Kazakhstan, in Stockton, Illinois, all these other places, that there is definitely that carryover. It is a compounding program, so each year it should get better. And then realistically, Ed, you can speak to this probably better than anyone on the planet, but then the genetics should get stronger and stronger each year in the hybrid.
Well, I don't know how much of the base genetics changes, but it does. I think you can adapt or collect more endophytes in the seed and over time. But or if you're using the same hybrid on the same farm, years ago you'd start to see hybrids get poorer performing, and that's why you'd switch out to a new one.
Sure.
And we always thought maybe it was genetic improvement. But now I'm beginning to wonder is maybe our soils are degrading with the way we're farming, and that's why hybrids lose performance as much as we were seeing genetic gain. I mean, there's genetic gain going on. Don't misunderstand that.
Right.
But some hybrids last longer than others, and my opinion is becoming they were better farmers of the microbes. We never gave that part credit.
The hybrid itself, you're saying.
Yeah.
Yeah.
And so the the hybrid itself was farming the microbe because my education was basically soil's a growing medium.
Right.
You put this amount of the components for your yield goal and boom, it's there consistently if you have the water. Well, now that my eyes have been open, let's say in the last 18 to 20 months, it's a whole different ball game, and so I think we're selecting the microbes that work with our hybrids when we breed under certain conditions, and the high stress conditions did that, and so I'm sure they're keeping those endophytes inside that seed, and then they take it wherever they go. And so the one thing we're trying to do in our seed production now is no fungicides applied. I don't care because we don't want to hurt those fungal endophytes that we have in that plant because we think that's critical in the F1 hybrid that the farmer grows. And so in the nursery field that Travis talked about, that's been no-till. This was its third time that we planted into it no-till. And so it's starting to build, the mycorrhiza and the fungi that we want just from stopping the tillage. And so I find it interesting that we found, a fungal grouping that was unidentified or unknown. We didn't apply it or anything like that. It's
Right.
in the soil and it was woken up.
Right.
How we're farming. And so that's kind of the undiscovered country that we got to keep digging into. What's the interaction between genetics and soils and how much do we carry with us? And so it may be our long-term game plan is just feed the microbes that exist. Maybe we start out in the farm, okay, we know these will help you get started, but after maybe 2-3 years, let's just go to feeding the microbes that you have. And
Sure.
that takes a different strategy on fields depending upon how long they've been regeneratively farmed.
So how do we, Travis, maybe you answer this question, is how do we go about that feeding process? What would be the method there?
So I think the root exudates is kind of the obvious answer there. Once we start going into a monoculture, we got one type of hybrid out there producing a particular type of root exudate. Just something as simple as mixing up hybrids, since they do have different exudates, cover crops, and having a diverse cover crop out there. That's all kind of simple ways to be able to increase your biodiversity and increase the amount of microbes. Then you can start getting into products, right? You can go into, okay, let's put out a fish hydrolysate. And the fish hydrolysate is going to have a lot of complex fats and proteins and amino acids, enzymes that support fungal growth. So if we need to boost our fungi, maybe that's what we're going to use, or a humic acid that is also going to be able to support microbial growth. So there's sugar. a lot of these things are out there, and we can experiment with them, and they all kind of boost different populations at different times of years. But ultimately, when we're shooting for diversity, being able to do that with the plant roots themselves is probably one of the easier routes to go.
Well, don't you think, Travis, that, the diverse cover crop can help explode that microbial population with a little bit of that food too? Because you've got, maybe 10 different species out there planted between the corn
Mhm.
rows and exponentially grow the microbial family we're looking for.
Absolutely. I mean, if you start looking at what we have planted out there at the Discovery Farm, we have things that like brassicas that are known for having relationships with bacteria, less so with fungus. But then, we start looking at a plant like flax, and flax is actually one that is known for boosting mycorrhiza populations. So by having that diversity in the cover crop mix, we're kind of trying to cover all the different bases, and it does great for building soil, organic matter, and water retention, and all these things that cover crops are known for. But I think much fewer people talk about how important it is to have that mixture of root exudates so you can start making a whole party of microbes out there in the field.
Oh, I don't know. I look at it where if we have all of those things in that field together, all kind of speaking that same language, and all working towards the overall health of that entire operation, that when we get out there, and even if whatever our treatment is, and it just makes that more powerful, and anything that we add to it. But I like the idea of waking up that biology that's out there, and instead of applying or over applying or doing other things, but mainly is to reduce the usage of things that are going to destroy that. And the more that we can do that, or not do that, I guess, is really where we're going to start seeing a lot of gain. And then, like you're saying, where the genetic gain is able to understand the language that everything in that ground is speaking, and then all working together.
Mhm.
Yeah, and one other thought now, after going through this process now on the Discovery Farm and continue to look at other regenerative ag farms, is you're talking about diversity of the cover crop. I'm starting to think about diversity of the F1 seed in the row, where we might pick some diverse genetics and blend them together. We'd still be planting F1 seed, hybrid seed, but it'd be maybe 2, 3, 4 hybrids put together of similar maturity that would create different root exudates and then attract different microbes. Because the anecdotal evidence for this is that when we do our research work, and so we have to put down a certain number of seeds to make sure the seed disc fills and plants out our 20 or 40-foot row. And then we vacuum out the excess seed when it reloads with the next hybrid as we're going through the field. So we get a mixture of seed then in the evacuation tank. And so when we get to the end of planting a plot, we'll just plant that seed on the borders of the field where we have some extra space or something. And almost always that fruity, what we call fruity pebbles mix, has all kinds of different colors of seed treatment in there from the various companies, out yields any individual hybrid that's out there. And so I think our work going into next year, we're going to be creating more mixes of F1 seeds to see if we can find that. Let's say a Magic 105 mix and a Magic 100-day mix, and I shouldn't say magic, but you get what I'm talking about. The ultimate mix.
So, I mean, our major concern then, if we went that route, would be just to make sure that the different hybrids that are in that would have similar dry down, or finishing out around the same time.
Well, finishing out about the same time, you want a little bit of diversity between flowering dates and times to spread your pollination time out. I mean, if we're going to do this, let's put a couple, 2, 3 hybrids in there that pollinate at different times. And then maybe with your latest one being maybe the best pollinator of the group.
Sure.
So it finishes well.
I like
So
that
I mean,
idea.
not all of them are going to have tassels like Vanessa does. So, but other hybrids may have a little bit better dry down than Vanessa. So overall, you when you blend the seed together, you'll probably be fine with the dry down, but your yield level should be higher just due to there's a zinnia effect. When pollen from one plant pollinates another unrelated plant, you get about a 2% bump in yield. And so with the mono culture, single F1 hybrid in the field, we're probably leaving a little bit of yield on the table. You know, but when you look at farmers have been splitting planters for years, but the issue becomes is what happens if one goes down and the other one's standing or the maturity is too different, then it becomes a problem.
Right.
And the one thing that I think we can see is that if we're not separated out by row or mixed together, those seeds will help each other more often than hurt each other because their roots do intertwine. So anyway, that's just some thoughts, So like I say, this Discovery Farm opens up all kinds of thought processes. And as I've been sitting in the combine so far this fall or traveling to look at field days, you start, okay, what does next year look like?
Right.
But the final piece of the puzzle is what's the combine going to tell us?
Sure.
And then we can take all these ideas and rank them in order of importance to work on, or if they're still valid thoughts or not. So
Yeah, now I'm thinking crazy ideas. So
I was going to say either we overloaded you or
no. I'm
pretty
thinking
fast now to see what.
Just got to find some people
It
that
is.
that are like-minded to run with some crazy ideas that I have running through my head. But they're out there though. I personally I like the idea of it, and I see it where we have plots that overall, if we have 8 varieties in a plot, that they seem healthier in that collective, even though it might be 8 rows of one and then 8 rows of another, but just collectively they do seem like they're, when you walk through there, or whichever variety you're going through, seem a little bit better than the field that might surround it or a neighboring field. So that effect that's happening is like you talk about, that there is something that's positive that is happening, to the point where in any situation like that, I haven't seen a negative impact from it at all.
Yeah, and pollen does flow. Corn is a cross-pollinated crop,
Right?
And so, I mean, corn's going to move at least 10 rows. Pollen from a corn plant will move at least 10 rows. And I mean, when we're isolating for seed fields, we need to be 660 feet apart, to prevent cross-pollination. So I mean, it's known that the pollen travels a distance.
Right.
Anyway. But if you have it mixed up more, with like within the row versus a few rows over, you're going to get more cross-pollination than you would just by splitting your planter. So from what you've seen in health, Andrew, with let's say an 8-row plots for show plots, it'll increase if you put the blends here. It'll get even, I think you'll see a noticeable improvement in health. Now, have I been brave enough to do this? No, not really. I mean, because you got to understand, how are we going to market this, and what are the seed laws that we have to follow?
Sure.
If we would blend hybrids together, and so we got to do a little bit of homework on that. I believe we can call it an F1 blend, but I'd have to reread the seed laws, or maybe we'll have Rachel reread the seed laws, because she follows everything to the letter of the law. Where
Right.
I'm maybe not so strict on that,
but also if an operator out there said that they wanted to go through it and they had, that they can do it on their own operation and don't have to worry about any of the stuff that we have to worry about.
Exactly. We can sell them hybrids that they can blend together on their own if they want to, or not. If a guy has a row planter, you could probably put a different hybrid in each row and be fine.
Right.
And just rotate through. But that these center fill planters, which is very convenient.
Right.
Then you got to figure out how you're going to blend it before you put it up in the hopper.
Right. Yeah, and you could still have a pocket of one just based off of the randomness of it all, and then a pocket of another. But overall, in that field of whatever you had in the center fill, it would still be very, very, you'd have more of a checkerboard effect than anything else.
The one thing that would happen, though, I mean, for a grower, if he's very stuck on having very uniform fields, this isn't the program.
No, it's going to look different for sure.
You look right,
Yeah.
different plant heights. And all that's the diversity,
Right.
and that's the key to what we're talking about in regenerative ag is diversity.
Mm-hmm.
Well, I think if you went down that path and you had a couple variety that were out there that were scattered, right, that were blended together, because those plants are all different from each other, and you have enough genetic diversity within that. Say you had 4 hybrids, and it had 8 parents, or we're increasing the quorum effect in that field. So I would guess that everything is going to increase in terms of availability for the opportunity for increased plant health,
Mhm.
disease resistance, all these different things that will increase. Um, I mean, we're seeing it a lot across the country. You just drive by certain fields, and you see that rust and that tar spot in certain areas has really been devastating across parts of the country this year. Um, I've seen it. It's bad. I've seen it, but when I walk through some of our stuff, I'm not seeing it at the level of some neighbors. That's for sure. So something's going on. That's a benefit. Now, some of this is I'll have to figure out a way to put this down on paper somehow, but areas where I have stuff out there in the country where there is standard treatment, industry treatment, compared to whether the operator put something on themselves or we did some custom treated or had biological seed treatment on it, those plants all look better than the hybrids that they had that were planted with chemical seed treatment. Healthier plants right now, more resistant to what's going on. But then on top of that, with all the things that are going on, if you do have some fields that are out there that have tar spot or rust that are in there, that stuff will overwinter. So there is a lot of importance on doing some sort of residue management program, whatever it is, whoever you use. You can get stuff from us, you can get it from whoever, but it's super important because that will help mitigate any of that stuff from happening next year.
Yeah, and so after harvest, Travis and Rachel have been putting together a stubble digester trial, and so we'll apply that as soon as we get the crop harvested, and so then we'll be able to study what advantages we had from that next year. So the whole idea of Discovery Farm is compounding itself to, I think every idea we get, we're opening up another rabbit hole to run down. But
Someone's got to do it, right?
that's what we do.
Yeah, someone's got to do that work because I don't think there's a lot of other people that are out there that are looking at it the way that we are. That's for sure.
Yeah, and it like I've said before, we've been breeding corn for this type of system for 20 years and didn't know it until 18, 20 months ago. And all of a sudden now how do we refocus our thinking a little bit? So, and then one thing I didn't mention about the Discovery Farm is now we've put every hybrid in our pipeline through skip row work to look for flex, and then also with cover crop interseeding. Everything is going through that.
What was the standard planting rate that we ran up there at the Discovery Farm in the non-skip row work?
So the population work was 34,000 is what the
30.
planner was set at.
Okay.
And
So
you're going to be a little bit from
we'll
that
see
base.
it. We're going to know which hybrids are going to, and have to, hybrid to hybrid on what that flex capability is going to be with those being our baseline numbers.
Mhm. Yeah, and so since I was planting into a cereal rye, I had the population at 34,000. I'm not sure that was necessary. I'd probably plant it a little bit lower. I mean, the standard rate for the county we're in as a Discovery Farm is between 34 and 36,000. But we have a lot of 22-inch planters, and I'm using a 30-inch planter, so 34 would be on the high side for us. But I thought I'd get some stand loss because of the cereal rye and the discussion about allelopathic effects. And I'm not sure I had enough cereal rye out there to get a full-blown allelopathic effect. So, I mean, we're kind of talking about what we saw here this year, things that I'm still questioning before harvest. That's another one of them. I really didn't see a negative effect from the cereal rye, except for the fact that I didn't get it killed with my herbicide when we sprayed it because we got a rain within, a few hours after we applied it,
Right.
to reduce the effectiveness. It was too wet to go spray again until the corn was up. So then I had no options left to control it. So then we had to go to the in-roll crimper, which I think saved us out there.
Sure. So, all right. So I'll ask you guys one more question each. Like out of all of the things that you've seen through the year at the Discovery Farm, and knowing that we're shortly, we're very close to combine time out there, is what is like one wild guess that you have on something that you think you'll see when the combine runs through there, to a hybrid or something? What do you think? And I'll hold you to it because we'll talk again after harvest to see if that wild thought carried through.
So I'm thinking that we're going to see a response from Dot in the skip row. Just visually going out there and looking at how the cobs are looking, how it looks like we have a larger seed set on there. Some hybrids respond by trying to put on double ears or a longer ear. She seemed to have gotten a lot fatter. So I'm really interested to see how that translates into total yield.
Ed, you got any wild guesses on what's going to happen when we see the combine roll through there?
Based on what I've seen so far, is we're definitely going to like the biology treated seed over untreated seed or over chemical treated seed. Now we don't have in our seed trials on the Discovery Farm where we had the big long strip plots, that's only naked versus biology, but I've got other seed treatment trials within the research portion where we got chemistry mixed in.
Right.
And I'm also going to say that I suspect the chemistry will be the lowest yielding of the three options. That we've been seeing that for 3-4 years, so it's a pretty safe bet for me.
Yeah.
Say that.
I think that we're doing things that are proving that we don't need to rely on that type of a treatment. Now, not to say that there are certain issues that could happen because of it, but then if we have tools in our toolbox that are available to use when certain things pop up, as opposed to relying on them every single year, I think that's pushing us in a positive trend. And the fields, the soil will be thankful because of it.
Yeah, we're pushing Travis pretty hard to keep researching what biology we need to do certain things so we can get it sourced and brought in and tested.
Mm-hmm.
Well, what I saw, and I guess this is what I'll leave it at, is I think anything that we had out there and what I've seen, whether it's stuff that I've grown myself or other areas, is that anything that has some sort of like the mycorrhizal inoculant that's in that seed treatment, that stuff seems to have definitely started off where it was really visible up to about V4, V5. So I think if we're getting ourselves set up properly up to that point, especially when we're going to be setting what the future of that year is going to look like, there's something to that. People should pay attention to it and maybe mix that into their operation because we need that plant up to that point to be really, really strong and healthy. And if we can increase that any way possible, it's going to be better this time of year when we're running a combine through there.
Well, the good thing is Andrews will be able to prove that with data.
Right.
Right,
Exactly.
It's not someone just say, here, try this, it works. It's, here's the data that shows that it works.
Right. Well, the one thing, was a message that I got a couple days ago where they had run a trial with a biological seed treatment versus standard 1034-0 starter. And he said, yeah, the 1034-0 is a starter. So things came out a little bit faster, initially looked a little bit better, but the biological seed treatment caught up very quickly. And then it just looking bad. He said, yeah, well, it's, it's not a starter, it's a finisher. And that's what we want. We want to be able to maximize all of that.
Yeah, no, I love that commentary.
Yeah.
And when we've done seed treatment trials and you'll see that the chemically treated seed does come up nice and uniform, and the biology treated seed would be a little bit uneven. But at the end of the year, it's what's in the grain tank.
Right.
And a very, very high percentage of the time, the biology treated seed wins.
Right.
Not 100%. It's almost there.
Right. Well, if we saw it in any of our data that ran out there that said we were leaving 30 to 50 bushel on the table because we were running biological treatment instead of chemistry, we would tell people. And it's just not what the results are showing up to this point. So
And I can only say that on our hybrids too.
sure.
That the untreated seed competes very well with the treated seed because we develop everything without seed treatment on. And so it makes sense that we could be hurting our seed by putting chemistry on it when it's used to running naked, let's say.
Right.
And then the
It's like
bottom,
dropping you off in a foreign country. You don't know the language. So it's going to take you a little bit to kind of get around and navigate.
that's a very good analogy, actually.
So
Very good.
if we can keep it going and know that we got those hybrids are, and not that ours won't work in a more traditional or I guess conventional system. You know, a lot of them will take whatever you want to throw at them and drink it up, but some of them don't. So, you know, just finding the right ones that are out there that are going to work in the system.
And we
Easy for
did
me.
do a lot of discussion here about the regenerative ag, but these hybrids are tested in
Sure.
all kinds of systems, so we
Right.
know how to place them.
Right.
So we know they're going to work in a more standard big ag system, or they're going to work in regenerative ag system, and as we learn more and more, we can get more specific about which hybrids, let's say, lean one way or the other.
Sure.
If there's a difference, that's just going to take a little bit of time to get that data set built.
Well, I
That
think
we're-
it's easier to take a hybrid that is geared towards a more regenerative style of agriculture and put it into a quote unquote conventional system than going the other way and taking a
That's-
purely something that was developed in a very sterile environment, if you will, of perfect input placement at through the development, and then putting it into a more regenerative system. I think it'll struggle as opposed to the other way. Is what I'm saying.
Correct. Because I mean, just like I said, we started out putting a lot of stress on when our products develop them so that they can handle whatever Mother Nature throws at them.
Right.
And so they can get into the conventional ag system and be fine. That was the intent, but we're finding out that they can really excel when you get into a more regenerative setting with biologically active soils. And that's where Armin comment is you don't know what you have yet for genetic potential.
Right.
Because you haven't been in the kind of soils that you need to be into to understand that
Yet.
yet. And hopefully we're developing that, and we'll get an opportunity in the future to bring on another farm and start this process over and see if we can speed it up. The conversion from
Right.
standard farm soil to highly productive microbial based soil.
All right. So combines will be rolling here pretty soon. I'd like to follow up and then just kind of go over a run through. We'll collect everything and get it all in front of us to take a look at. Did the observations that we were looking at through the year, are our results in line with those observations or expectations? Where and then all that does really is help us talk to people in the future on selection, for sure. And really be able to hone in on, the right set of hybrids for an operation, which is exciting. It's super exciting because then it makes me not be able to not sleep at night because I think I picked the wrong hybrid for somebody.
And we don't want that happening.
No.
So we're going to more and have more information for you to draw from when you make your decisions.
Yeah, sure.
When you're working with it.
So Ed, you're next for you. You're headed to Kazakhstan soon.
Yes. About a week from now, we'll be headed to Kazakhstan.
And then we'll be rolling. So what was the guess again? When do you think we'll be in there? Like a week or two out there?
I'll be there Monday next week.
No.
Right?
I'm saying when in the Discovery Farm, when we'll be combining that? Like
Next week.
next week?
Yeah, we've already started combining other locations.
Okay.
And the road, and we'll be on the road next week, plus, here and then on some other locations in this area. Plus, we'll be at the Discovery Farm. We have multiple combines and crews, and so we attack a bunch at the same time. So we're red hot and rolling.
Well,
So-
yeah. Then once we get that and we look at the data, then let's have another conversation about what we talked about now, but in relationship to what the results were, good and bad results, whatever it ends up being, right?
Yeah, I saw my first little bit of data this morning, and it's very exciting.
Okay.
It was in conventional farming, but it was with lines developed in the regenerative ag system.
Okay.
And so they're performing there. So I can't wait to see what they do now on the Discovery Farm.
Sure.
But it's really, this is cool. This is the time of year we wait for. We go work all year to get this information.
Oh, yeah.
So,
Well, I mean, that's all I got, unless any of you guys have anything else right now, last minute things here to bring up. But if not, then we'll revisit again after we collect harvest data from the Discovery Farm. And then
so yeah, I
plan
think
together.
one other thing, Andrew, we can mention to our followers here is that we will be publishing this online, what our results are. So that we want to be very transparent about what's happening and what we're seeing, and then, be an information base for us and for our customer. So they can make decisions on their farm.
Yeah. And once we have all that out there, we'll make it very easy to get to and let everyone know where it's at.
Yeah.
Well, thank you guys. I appreciate it. Thanks for watching, everybody. We'll see you next time.
Thank you. Take care.